From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on August 22nd, 2024. Hello, Micronauts, and welcome again to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 55th episode of Matters Microbial. Thanks so much for listening and/or watching and for spreading the good microbial word. I hope to make it to 100 episodes and more. Again, thanks to you. I wanted to now quickly tell the story of the term micronaut. When I teach microbiology to students, I strive to find ways to incorporate creativity. The students who claim to be the least creative often amaze me given the chance. So in the fall of 2016, I had a student named Mariko in my microbiology course. She often would doodle on exams in pencil and seemed quite artistic. Here's an example of how she portrayed the microbes living in her water bottle, the water bottle buddies that I so often explore with students. Her work was really charming and amazing. For her final creative project in my microbiology course, Mariko created this wonderful thank you illustration. Now, at the time, I used the term microbial supremacy. Sadly, that last word has a bad odor nowadays, so I changed it to microbial centrism. The overall thank you illustration has many comments, diagrams, and quirky things that I say in class. For example, I often stated that students were petri dishes with shoes, and that apparently really stuck with Mariko, as you can see. But at the top, her interpretation of the term micronaut I would use in class amazed me. I did use the term micronauts often to students in lecture, but she added "we soar" to the term. I mean, it's pretty amazing when you look at it. So I asked Maggie Middleton, a fabulous scientist and science artist, to work from Mariko's illustration. I then had it turned into an enamel pin, as you can see. Here is Mariko posing with one of the pins. So now you know the story of micronauts. When you hear students tell you that they lack creativity or artistic talent, please remember this story, Mariko's work, and encourage them. I am very lucky to know a large number of microbial enthusiasts from the American Society for Microbiology and from the educational wing of that organization. Many of us are very interested in promoting microbiology as much as we can. I find every year that there is only a small amount of microbial information in introductory biology. One of the most stalwart and prolific defenders of the one true microbial faith is Dr. David Westenberg of the Department of Biological Sciences at the Missouri University of Science and Technology. Dave is also a Karski Awardee for Excellence in Undergraduate Education from the ASM, has given a TED Talk, and has an endless number of activities and accomplishments to go over. So, Dave wears more hats than I do, and is above all a kind and positive influence on many people, including yours truly. I'm honored to call him friend. Dave, welcome to the Quality Quorum for today.
Hey, Mark, great to see you. This is exciting to be on this, and I'm going to start off with congratulations on over a year with this podcast. I'm so excited to see you accomplishing what you set out to do. So I'm really proud of it, to be part of it.
Well, I'm delighted you're here. Lots of different types of people have been on the show, and as I've said many times, this particular podcast is not simply informational for others, but so therapeutic for me.
I hear you. Watching them have been great to again, that energy, excitement is so cool.
It is nice to hear you say so. So what I like to do when we first start is talk a little bit, and you described yourself to me previously, and I love this term, an accidental microbiologist. And I'd like to know more about that particular journey that took you to today.
Yeah, absolutely. Yeah, I was, kind of a late bloomer to the microbial world and the microbial centrism. When I went to college, I'm a first generation college student, just like yourself, and you didn't really know what to do with, you love science, but what kind of careers can you have in science? And so my passion at first I thought was to be a veterinarian. So I went to college to be veterinarian, but I wasn't really cut out to it, but that was always, you talk about all these years. When I got to college, I realized you can't major in pre-vet, right? You can't, what do you do if you don't get into vet school? So you have to pick a major, right? So I'm going through all the options in biology and microbiology. Oh, this looks interesting. Again, for all the wrong reasons. I saw, okay, well, I want to study animals. They're going to get sick. I want to learn about diseases and that stuff. And so I, picked microbiology as a major. And as you all know, when you get into that microbiology class and you see the incredible diversity and the impact of the microbial world, that just profoundly changed my view of where I wanted to go. But then still didn't know what to do with it as a degree. So I kind of went along, and I loved my classes. I had some great classes like microbial genetics that was exciting. But what really solidified things was, as I tell my students, my last quarter of my last year of undergraduate, I took a microbial diversity class with John Bresnak at Michigan State University. And I got to do basically like a cure. The thing that's really hot these days, 40 years ago, John was doing things like, let's isolate bacteria and identify them and characterize them. And so that was like, I can do this. This is exciting. John had great encouraging words. I still have my last report somewhere in my folder here where John had very kind words about, you should look into going to graduate school, and now you've learned about these diverse microbes. Maybe apply your interest in genetics to diverse microbes. Don't be another E. coli geneticist was the way he worded it. And so those kind of things stuck with me. But that's when I really realized, hey, I can do this. I can become a professor. So for me, in my career, I've always kept that in mind, that it took me so long to get to that point. I want to give students that opportunity to realize early on in their undergraduate days that there's more to life than med school and vet school and things like that. There's all these great careers you can get in the sciences. And so give them that exposure to the things that I got exposed to earlier on, rather than waiting to that last quarter, the last semester.
Hey, Dave, did you ever think it was strange that microbiology was required for veterinary school, but not medical school?
In a way, and ever since I've been an advisor of the pre-med students, I get it. It's a very interesting reasoning, but yeah, the vet schools are much more rigorous in their expectations of preparation for these things. The med schools always found interesting. The requirements are actually quite limited. There's not a lot of required classes for med school. So I've always kind of found that very strange. But at the same time, when my students go off to med school and they write back and talk about how, they appreciated my microbiology class because it helped prepare them for their med schools. But I think the way I've seen it and the way I've heard it is that because the med schools, they want to teach you their way.
Yes.
If you come in with too much pre-knowledge, you're kind of biased in one way. They want to teach it their way. That's the way I seem to see it.
I think you're right, but it always amazed me, and this is again from discussions with students, that microbiology is required for ophthalmology or optometry. It's required for veterinary school, pharmacy school, but not medical school, which and dental school, I left that out. So it's very interesting about that. But what you said about medical school and those listeners and viewers interested in medical school, I don't envy you, because it's a shifting target all the time. Best of luck.
Yeah, it's highly competitive. And again, I always have to remind students that not getting in isn't something against you. There's a lot of qualified people that don't get in. They're looking for particular things, and so it's always important for me when working with our students. I love our pre-med students here. They're great kids, but sometimes you have to realize that there may be other options they need to explore, and so I like to give them those options to look at. And so proud of them, whichever direction they go with their careers is always kind of cool to see. But when I reflect on my time, I think about, my mentors. And so I want to be that kind of mentor, just like the kind of mentor you are for your students. We take the same kind of pride, I think, is when we see our students go on and be successful. That really makes us incredibly proud. And not that, I don't think I do anything magical, but I just feel like I gave them that exposure that they need. So.
We are just talent scouts, Dave.
Yeah,
just like
exactly.
John was a talent scout for you.
Yeah,
and that
exactly.
that thing that he wrote had a huge influence on you and encouraged you on your path. So you went to graduate school.
Yeah, exactly. And I got one little piece of kind of like a reminder of that time period that I've kept with me all these years as a reminder. Taking John's microbial diversity class, one of the prized possessions I have from that is a Carl Woese pin. So he was friends with Carl Woese, and so we had these buttons that Carl had made, and so those were our rewards for isolating our microbe in the class. And so I keep that with me today. It's on the wall right next to me. So it's again, having those little reminders is always kind of cool along the way. But
I'm really glad I'm glad you shared that image, which is visible now.
Yeah, isn't that neat? The 80s belong, kind of dates us a little bit, but it's good to have those memories. But John was a powerful influence. And so when I was, this idea of graduate school, what do you do? This is the days before the internet. So, sending off remember that day, sending off little cards to the schools and getting information and where do I apply? And John was a great resource for me, encouraged me to apply for places I hadn't, I was always, didn't always challenge myself as well as I could have. And when I looked at grad schools, I looked, okay, what can I get in? And he told me, you got to look a little bit beyond, go for those safe schools, go for some that you think are a good fit, but also, go for that pie in the sky. You never know where you go. I mean, I never would imagine I could get into UCLA as a PhD student. So getting that opportunity was, again, thankful for him to really pushing me to put myself out there and challenge myself a little bit. I know that's something that can be hard for us to do. And that's, again, this accidental microbiologist is a lot of things I've accomplished are sometimes out of sheer ignorance, about how challenging it was. I might not have ever done it if I realized how difficult some of the things I did. Say, getting into a great PhD program led me to a postdoc over in Germany that, again, with Gerhard Gottschalk, I mean, who could have dreamed getting into a lab like that, getting an Alexander von Humboldt fellowship. I applied for that with absolutely no clue how competitive that was. But it was that encouragement of others that helped me to get there. So that was very cool.
But I know your PhD advisor quite well. He was a postdoc when I was a graduate student at Stanford. And minute I saw that, I knew you'd be properly mentored, because that's really important, is to have somebody, just as you learned with John, who's able to kind of evaluate and encourage and kind of balance, because a lot of times we don't know what the next step is. And that's one of the things we've talked about on the podcast. They're like, all roads lead to Rome, as the saying goes. Well, I would say that there are many paths to microbiology.
Yeah, absolutely. And again, just putting you in those opportunities to let you make those discoveries on your own, kind of find new directions. Working in, I went into my lab for the PhD because John introduced me to the methanogens, the archaea. This was an early day. I wanted to apply genetics to methanogens, studying archaea, but we weren't quite there yet. And so I working with Rob Gonzalez, great pedigree. He was also had the background in the genetics, the bioenergetics, but also the methanogen background with his days back with when he was at Illinois. So a lot of great ties there. So I ended up being, again, I don't follow advice very well. When John Bresnik told me, don't become another E. coli geneticist, what did I become? An E. coli geneticist. But I was an E. coli geneticist that was at least studying something unique about E. coli, anaerobic respiration was not the traditional way we think about respiration. And so an opportunity to work on that kind of a different side of the E. coli story was fun to do. But the connections being in Rob's lab and getting to meet Gerhard Gottschalk, and saying, hey, I'd love to come to Germany and do a postdoc. And he said, yeah, apply for this fellowship and see how it goes. So those, again, surprise, accidental connections really send us in interesting directions. So it sent me to Germany for a couple of years. So that was a great experience. My daughter was born over there, so a lot of great fond memories of that time. So that was good. But then I had to come back to the States. And there was another adventure. What do I want to do for my next postdoc? And again, learning about, I think you had these on here, some of the heavy metal stuff. The metals was fascinating to me. This idea of my fascination with the bioenergetics and anaerobic respiration, all these different types of respiration. And that was about the time when folks like Derek Lovely was making all these great discoveries with geobacter and breathing metals and things like that. So I reached out to all these metals people. And again, this is the days before the internet was really taking off. So I'm sending letters to to these folks, and then waiting for those letters to come back. Some of my letters got crossed in the mail. Sometimes I put them in the wrong envelope. So all these little things that came along the way was kind of an adventure. But eventually ended up again in again one of these accidental opportunities. The ones that really where I thought I was going to go again into this metal respiration world wasn't paying off, and so I found a lab at Dartmouth with Mary Lou Garneau that was studying iron metabolism. I said, well, iron's a metal, right? So some interesting aspects of iron metabolism that look fascinating, and so I took up the opportunity and had then 4 great years with Mary Lou learning about plant microbe interactions, and so it sent me in a little different direction than what I originally imagined. But I think that's important when you're going through these paths is keeping your eyes up, looking for those opportunities, and you never know what's going to fall into place. And so again, those small little things that can steer us in the right direction. So that was a great experience.
2 observations, Dave. The
Yeah.
first is, the OG of microbiology, or one of the OGs, Louis Pasteur, used to say chance favors the prepared mind, and that's absolutely true. The second, of course, is the Grateful Dead. What a long, strange trip it's been. And it's really true. And especially when you're first starting out, you're sure that you're moving from A to B to C to D. It's actually much more, and I hate the term drunkard's walk. We'll just call it a random walk.
Random walk.
And we end up to a place where it's a really good fit. But it takes confidence and faith and all kinds of things to get there. And most of all, a supportive group. And it sounds like you've been really lucky in that regard.
Absolutely.
So,
I've had great mentors.
So that took you now to a particular job, and the job that you have at Missouri, you're doing a lot of different things. And I think the first thing that you wanted to talk about with me, and I think people will be interested in hearing about, is the huge effort that you have in outreach, and you always have for as long as I've known you.
Yeah, that's the thing that actually, again, it was kind of that transition. When I was at my postdoc, and I had lined up a faculty position here at Missouri, at the time, University of Missouri Raw, we were called, but Missouri S&T is a newer name to kind of fit in with our engineering world. But I went to ASM because I figured this is an opportunity to start planning for my new lab. And ASM that year was in Atlanta, and they were doing an interesting thing they called Teacher Science Day. And I said, okay, what's this all about? And so ASM, what they did is they brought teachers from the local community. So this is Atlanta. So they brought in some local K-12 teachers to spend a day at ASM. And they had scientists that were doing activities with them they could learn in the classroom. And I thought, this would be, cool thing to do. Now I've got family in Atlanta, so I felt this connection. I thought I'd like to learn about, maybe I could help my family in a way. And so I attended that science day, and that opened my eyes to the power of working with the K-12 educators. And I say this unabashedly, my inspiration, everything I know about teaching, I learned from K-12 educators. I have such profound respect for the K-12 education community and what they do and the challenges that they face, and finding ways that we can support them in their endeavors. And so that got me introduced to a lot of K-12 educators and some ASM members that were working with that community. And so that opened my eyes. So I immediately said, okay, when I get to Missouri, again, a rural community where, again, a lot of our schools in this area don't have access to a lot of the resources that they could benefit from, I thought that would be an impactful thing that I could get involved with. And so my kid was just getting into the school, so that's the perfect opportunity. Okay, there's a science fair. I'm going to do some microbiology activities. Her class is talking about some topic that I could draw microbes into, and we all know we can connect microbes to everything, right? I think I had to hurt my department chair a little bit, because everything we talk about, I draw the microbes into it. And so let's bring microbes into the classroom. And so so I would come in, I'd bring in books about microbes, I'd bring in things to show them. And I knew it was having an impact. One summer, my daughter was in youth soccer, the typical, the little huddle of kids moving across the field, and standing in, one of the parents kind of came up to me and said, "Are you the germ guy?" Apparently that's the reputation I had with the kids, that the kids called me the germ guy. And so, okay, I'm having an impact. The kids are remembering, they're telling their parents about it. I said, this is something I could get myself into. So then started seeking out more ways to do that. And as I tried to be creative, that led to different things. So, you do that great Lux art. My daughter's class was doing a class where we're looking at the ocean. And so I said, let's bring in some bioluminescent bacteria to see how that works, talking about those symbiosis. I remember a demonstration John Bresnak did. I still do this to this day, where he took the Photobacterium lignathii that you use, put it into a tube.
Oh, yeah.
And let it go dark. And then you could rotate that tube, and as the air bubble went up, they started to luminesce. So doing those kinds of things. So that's kind of how I then followed in your footsteps with the Lux art, to do that with the classrooms. So that was kind of fun.
Did you know what Bjerink used to do? He would take like a piece of seaweed. He would put Photobacterium into the seawater. He would shine a light on it very, very brightly. Now that's sitting there in seawater, and then he would turn off the lights. And anywhere oxygen was produced, the bacteria were using that to make light. And so you would see the outline of the leaf.
Very cool. I'd never heard about that one. That's cool.
Oh yeah, it's not super easy. You should give it a try, because it's really striking when it works.
That's challenging. Yeah, I still remember. You and I, we both did the microbial diversity class at Woods Hole. The year I did it, we had one of the students in the project took put the bioluminescent bacteria inside of a chamber, and then around the chamber put phototrophs around it, and to see if they could use the light to drive photosynthesis. And they got a little bit of oxygen coming out of that. How much of it's real, I don't know. It was a fun attempt.
Oh no, we've learned so much about deep photosynthesis now. I wouldn't be at all surprised you get a little bit out of it, but that's a perpetual motion machine there, right?
Exactly. Yeah. So those kind of things. Yeah. Finding those connections to get the kids excited, and moving away from that. Again, I use that word germ very affectionately, but trying to get them to realize that germ isn't always a bad thing. Trying to open their eyes to the positive side of things. But of course, they liked all the scary stuff. So, I still remember one year my daughter's class was talking about the Middle Ages, and I came across an activity about the plague. And so I said, okay, let's do this activity with her class, and actually got a little bit carried away, and actually talked to our theater department and got myself a plague doctor costume made up. And so showed up in her classroom as a plague doctor and took them through the Middle Ages. So, finding that way, you got to be, as parents, we got to find some way to embarrass our kids a little bit. So, showing up and doing that.
That's what all parents do. I wanted to say something about the word germ. This is a big deal to me.
Right.
Pasteur popularized it. And what he meant when he said it was a piece of life. Yet the word germ has all this negativity in most people's mind associated with dirt or disease. Yet most super health foodie people have a bottle of wheat germ.
Yeah.
So I'm going to have a photo that'll be posted here about what I think about wheat germ.
Yeah.
So my point I'm trying to get at is that it's all about microbial literacy, and it's all about microbial outreach. And you've done, I love the photograph that you sent about you working with a bunch of quite young people about microbiology. And it's good that you're spreading the word.
Yeah, it's fun. That germ of an idea that takes off. And, I it's not quite the direction I meant to go with my social media handle as the germ juggler. I usually tell people it's I enjoy juggling those two concepts. We got to respect the microbes. That's my take home message is always, most microbes are beneficial, but we have to respect the microbe. And so that idea of juggling these concepts that there are dangerous microbes out there, but they're quite rare. But we don't know who they are. So we have to juggle that concept of good versus bad forms of germs.
We took a photo of you and I, as the germ juggler. That I'm going to see if I can find a picture up to put up. That was really quite good.
Yes, I've got that. I've used that, and again, that's one of those that freaks out my department chair when I show that one. I love that one. We did two of them. You did one where we made the germ juggler go around my head,
right?
And then we have the one where we had the three of us with our heads in the bioluminescent. That was fun. I forgot I was going to add those.
Very Game of Thronesy, yes. It was a lot of fun.
Yeah, that was fun to do. So so outreach has always been a real passion of mine, and so one thing again at the time ASM was very much involved in that. So that kind of took me to get a little bit more involved with ASM that way. To kind of work with their K-12 outreach activity. That connected me with all sorts of new people. The USA Science and Engineering Festival was a blast. It got me involved with National Association of Biology Teachers. And then eventually I got really blessed to be able to work with the Howard Hughes Medical Institute, doing some of their work. And so outreach, sometimes I was criticized a little bit. You need to focus on your research. You got to focus on your bring in the big grants and stuff like that. But again, I'm a little stubborn. I'm going to do it my way. I'm going to this is more important to me, and so I'm going to do what's important to me to build those connections. And I always believe that generally things are going to work out. And so maybe maybe it's not the right thing to do, but the right thing in one sense, but it's the right thing to do for me. And so I'm making sure you stick with it, and it's paid off in ways I couldn't have imagined. So you have to do the right things.
I want to emphasize that to the listeners and viewers is that often we'll all get told this is the right way to do something. It doesn't matter under what context. I've had it said to me, someone who I just adore as a human being and as a scientist and as a mentor once told me that I should be a little less markish. And what's interesting about that is that she didn't mean anything negative, but what she meant is for her, it's you when you give advice to others, it has to be seen through that kind of prism or magnifying glass. I mean, what works for you might not work for me and vice versa. That's very common, but you have to find the niche for yourself that seems to work properly. So again, when my friend said, try to be a little less markish, that was simply her view. I actually spoke to Abigail Salyers, who you no doubt know was so influential to me. And she made a big deal about that with me, saying it's important to be who you are. And I can really underscore that authenticity is vital because everyone responds to authenticity. They might wish you had been more like them, but the question is, how good are you at being you? And you, Dave, have been really good at being you.
Sure. And students can sense that too. And
yes, they can.
You gotta be who you are with that kind of stuff. So it's been to me, that's been, again, that's what keeps me going, is having those opportunities and the opportunities that come here. I'm very fortunate to be at a university where, those kind of things are supported and recognized in a lot of different ways. We've got a lot of good people that have always encouraged me, even though they maybe try to do more of this, but they've been very, very supportive of that. You remind me of that. I have to keep that in context a lot. And again, it's an idea of being yourself that really helps draw people to me, I think. When I started here, kind of going in a little bit direction about some of the research opportunities I've had here, and I always have to catch myself with that. I don't really do anything. It's my students that do all the work. I'm making the connections. I'm doing that. It's my students that do these things. I find them, but yeah, it's all about the students. And so I got here, and people recognize that I'm someone they can work with. And so, you get these interesting opportunities. Again, the connection with the high school is also an important part of this. One of my first really fascinating collaborations was a high school student, reaches out to me and says, Dr. Westenberg, I'm doing this project. Can I borrow some bacteria? It's like, okay. You want to borrow some bacteria? Explain that to me. Come by my office and tell me what you're doing. And he had been working some stuff at the local hospital, but they didn't really know what he was doing. And so he said, okay, I'm studying some bacteria. I'm working with this ceramic engineer. And I think ceramic engineers, what do they do with microbiology? I'm thinking toilets, but they were working on glass for bone repair. Glass has a lot of properties of the bone. And so this is the guy that's doing this project, again, accidental things. He's a member of the National Academy of Engineers. How do I get to work with people like that? That's just mind boggling. But this high school kid was working with him on a project to look at this glass. And their idea was, well, if you've got broken bones, you're at risk for infection. So can we use the glass to help fight infections? So they were testing to see if they could make this glass kill bacteria. And so they were incorporating silver into the glass. Mm-hmm. And so this kid was just sprinkling glass onto a petri dish and covered with bacteria and looking for those zones of inhibition, classic antibiosis. And so I said, okay, this is cool. Let's see, I've got a collection of bacteria. Let's see if we can make a project out of this. And I've been doing it now for almost 27 years now, working with bioactive glass in different flavors and stuff like that. So again, it's been a really cool collaboration. It's a great project for undergraduate students because it's pretty straightforward, simple in one context. But again, it's things that they can really make discoveries. You talk about this all the time, that I particularly say that I'm seeing something that no one else has seen before. So they're saying, okay, this glass can do this. Here.
And it's interesting. A lot of times when there's a straightforward project, other people will critique it. And I'm here to say that at least my students understand every aspect of what they've done.
Yep.
And I'll give you a really good example. Yeah. That'll make you smile. When we talk about freezing bacteria at -80 to preserve them, I've never seen anyone look at it like I believe Rich Lenski did some work on this in another context, but how quickly do they die at -80? Because people don't check.
Yeah.
And it's the perfect kind of project for a student to kind of work with, to learn about the manipulation of the numbers, to be able to present their data, to be able to draw conclusions and ask questions. It's a great way to learn to be a scientist.
Yeah, absolutely. And again, sometimes having that student mindset also really benefits us. We got those blinders on. We got, this is what we want to accomplish, and the students are thinking a little bit differently about that. So a lot of those early students, okay, well, why are we doing it this way? What if we did it this way? Yeah, let's try it. Again, they're thinking about it. They start getting into, okay, how is this class actually working? And
yes.
It's an interesting thing. Engineers about, does it work? As a biologist, we want to know, why does it work? And so we complement each other very well. And it's, I'm really reminded of one of the first students on this project that came up with one of the ideas of, again, we were just sprinkling glass on, and so you really have no control of that. And so she came up with a way to compress the glass and make a little disc out of it. Very successful at that. We got some really great work. She went on to medical school, and to bring things full circle, her daughter's now in my freshman class.
Oh, how about that?
How awesome is that? to have those things come full circle like that is really neat.
I really appreciate the figure that you sent us about that, because it's a really good, I think, illustration, especially for people who are relatively new to the ideas of microbiology, about how to answer questions like that. So
Yeah.
thank you.
Yeah. And to get that kind of image, it took that student to come up with the idea, okay, how do we compress it so we get a consistent zone of inhibition around them, and then testing it in different types of glass. So we were able to figure out the composition of the glass and make that work. So that was a great experience to get, and kind of really got me connected. Okay, working with engineers is okay. They're fun to work with, and they come up with creative ideas to do things. They reach out to you for different ideas. One colleague was actually a pair of colleagues, in a mechanical engineer of all things, and a chemist. And Missouri's a big soybean growing state. So we have a soybean merchandising council is what they call it, that does funding of research. And I had some benefits. I was studying soybean symbiosis in my real job. And so I was very connected with that group. And so through my conversations with this group, said, well, we're using soybean oil to generate fog oil. So we're down the road from a military base, and the Army uses what they call fog oil to hide their maneuvers. So basically, they literally blow smoke. So the Army's blowing smoke, and they hide their maneuvers. And so their idea, well, they've generally been using petroleum oil. Well, what if we use soybean oil? A little bit more environmentally friendly. But they needed to test the safety. Okay, is this a dangerous thing to do? And so the one test we have as microbiologists is the Ames test, right? The Ames test for mutagenicity. And I have the collection of strains that can do the Ames testing. And so they reached out to me and said, okay, can you test and see, is this oil vapor safe? And so we started doing Ames tests. And the first thing that, again, this odd discovery, the student that was working at says, I don't see anything. Nothing's growing. We couldn't get any growth and any difference the way we set it up. And so it looked like this oil the vapors from the oil is killing the bacteria. So we tested against our whole spectrum of bacteria we have in our collection. It killed everything. Very strong antibacterial properties. Again, this is a few years ago. This was around the time of the anthrax scare in Washington, D.C. And so we could actually have this oil that we could actually fog a building to decontaminate, because it could actually kill bacterial spores. So again, accidental discoveries, through connections that we make with colleagues, and again, students got to do this work. I found the connections, I connected the students with the things we need to do, and they got to know those questions. Well, why aren't they growing? What's going on here? How else can we test it out? So it's been a fascinating adventure with those types of things.
And those are great projects.
Yeah, absolutely. They're geared for students. To be able to do that, to get them to think, because it connects to them in a way, and it's something that's unusual. They can make their discoveries. So that's some lasting projects with them, and probably the last project I think that might be of interest to you, that I again, thing that blew me away. What happened was, I had a student in my microbiology class, a chemical engineer, but we have a design team here called Concrete Canoe. They actually make concrete canoes, and they compete and they float their canoes. I just learned the other day that it's the earliest design team we had on our campus. But this student, he had heard my microbiology class. He heard about these wound healing, cement concrete healing bacteria. He said, can we get these concrete healing bacteria and put it into our canoe? Sure, why not? So I went to the ATCC. Turned out ATCC didn't have it. I had to go to the Belgian culture collection to get this organism. But I got it, and we had it in the concrete canoe, tried it, but didn't really know what to do with it. A few years later, a new colleague in our civil engineering department, he studies concrete. And one of the issues with recycling concrete is it generates a lot of really fine powdery material. And so he was developing ways to make aggregates out of it so it could be used to regenerate concrete. So he said, can you get these bacteria to do that? Well, sure, I've got it in my collection. Let's test it out. And so he had this time it was a PhD student in his lab that was looking at all the parameters of getting these aggregate formation. And what that led to is this spectacular electron micrograph that shows these bacteria as they kind of crystallize the calcium carbonate that's in there, and also creates almost like fossilized cells in there. When I saw those micrographs, that just blew me away that they can actually create a structure like that. Was kind of neat to see. And so again, that was a fun thing. We got a nice publication out of it, a couple of conference posters. And again, the thing is, we learned something new. I made some collaborations that I couldn't have imagined, and so it's been a cool ride being in engineering school. Most people think of that as being a disadvantage, but it's an advantage. You get to do things. They're kind of cool.
Thinking about those soybean oil clouds, Dave, I was wondering. I understand. I don't have any background in theater, but I understand that a lot of the smoke or the fog machines that are being used in theater are made from mineral oil in some fashion, and I'm hoping that's not that isn't an issue, because, or maybe it should be an issue, that it should be antimicrobial because of the possibility of transmitting things you wouldn't want to across the audience. Do you have thoughts?
Yeah, absolutely. The interesting. So a couple of points about that. When they do the smoke, again, it's the temperature. So we actually tested different temperatures for generating the smoke. And so there's like a lot of things, it's just a fine window to get the antibacterial properties. So we have to heat it to 600 degrees centigrade. That seems pretty high. But it's a very high heat to get the antibacterial properties. And so like the things they use don't use that much they're more heating droplets. So my concern is more like, the what do they call it? Popcorn lung, the fine oil vapors from vaping, the oils from, those, air fresheners and stuff like that, that use like what's the term they use for the oil that, No,
I know what you mean though. That's something that I haven't thought of. That's very interesting, Dave.
What do they call those types of oils that you use to create, again, to people use for like aromatherapy type stuff? There's a term for that type of oil. But anyhow, but yeah, I've got a colleague that's actually been studying. Again, I'm trying to get him to use the Ames test stuff, but vaping, because that's the big concern with vaping is it's like popcorn lung is a real thing, because if you inhaling those fine oil vapors can damage your lungs. And so is the vaping causing that problem? That's my bigger concern. I wouldn't be too much concerned about the microbial composition, more the interaction with our bodies. So, but it's interesting, the oil vapors that we have, we've tested against eukaryotes, and it doesn't affect mice or anything like that. It's only bacteria.
Huh. Interesting.
So, and my curiosity, of course, is what's it doing to our microbiomes? If you're inhaling this type of stuff. So what's that doing? But we had some interesting projects with it. We tried it out with Hurricane Katrina. We had some damaged buildings from that. We tried to disinfect. So yeah.
I really enjoyed seeing some of those great photos and images, photomicrographs of your concrete aggregates. And the idea of self-healing concrete is really interesting. And it kind of made me think a little bit about, as I have read about this self-healing Roman concrete. And I wonder if there's a microbial component to that people haven't looked at yet.
It's quite possible. That's something that would be great to explore. That's something to reach out again for my civil engineering colleague and see if he knows more about that. From our perspective, how do we build it and make it more durable? But there are some other applications we've looked at with these bacteria. I have another colleague that I was working with trying to look at things like sediment in rivers, and some of the impact that might have on bridges. And so they were looking at using this as a way to reinforce sediments so that you get less of that impact on bridges for the longer term survival of the bridges. And so that we were working with some creative ways to how to get the bacteria into these sediments. And so those are some of the engineering challenges that go behind it. And so again, that's the fun thing about working with engineers. They've got these ideas. Okay, we can make it work. So let's see if we can find a way to make it work in this context. So, but yeah, that's a great question. Is that part of it? Generally, again, we think of the damage that bacteria do, right? A lot of these stuffs are built around the damage bacteria do to statues and things like that. But are they also playing an important role in their durability as well? So, great question.
No, I think that, a really good example that comes up again and again, or not example, but a theme that comes up again and again on this podcast
Yeah.
is the idea that things really advance when you have different points of view that come together.
Yeah.
And I really believe that to be the case.
Yeah, I loved. I went to a talk by Rita Colwell one time when she was NSF director, and that was the point she made is life happens at interfaces. And that was her goal. Is that, and I've always looked for, is that quote out there somewhere? I've never been able to find a direct quote about that, but that's always been my mindset, is that interface of different disciplines is really where life happens, is always at the interface. So that's stuck with me a long time, to stick with that. That was a great way to think about these things. And so, yeah, looking for those connections that we can do things. And so, so kind of maybe one last thing to kind of bring into the conversation is, again, where that can take us in the future, with these types of things. And so early on in my time here, and again, credit goes to a colleague. And I always got to make sure that I want to make sure I recognize the colleagues that help drive some of the things. I'm kind of building on the shoulders of others. Again, the things I've learned from you, I've learned from people at ASM through other ways. I've described myself as the Milton Berle of microbiology, because I steal all my best material. Now, it was Milton Berle, that was his reputation. Famous comedian, but he was always kind of, you stole that from this person, so that one. Yeah, I bring them all together from different things. So, I guess that's an old time joke, I guess. Who is Milton Berle? But anyhow, a colleague from mine went to a conference and heard about this program called iGEM, International Genetically Engineered Machines. And this is a presentation by a local small university that had actually partnered with Davidson, of all things. So it was kind of a neat collaboration. And so this colleague at, I think it was Missouri Western State was the university they were at, talked about their experience doing iGEM with the Davidson iGEM team. And she came back and said, we need to do iGEM here at S&T. And so we looked into it, what it involved, and we got a team of students together, engineers and biologists, working on projects, going to the iGEM Jamboree, and getting to meet. What I love about iGEM is these students from all over the world. This is like a nerd fest, meeting other students from all over the world that have the same interests as them. And our kids are bowling with the team from Freiburg, Germany, and they're dancing with the team from Taipei, Taiwan, and they're all sharing these connections. They realize this big world out there. So I just loved having that connection. The joke, I got to get my dad joke in here that I usually tell the prospective students, every continent is represented in iGEM except for Antarctica. Do you know why? Penguins can't pipette. Okay. Dad jokes. I got to get those dad jokes in now, and I can't.
So I'm going to make sure that I get you an illustration of a penguin pipetting. That's what you need.
One out there. I'm sure there's one out there. But yeah, so our kids were really getting into it. They loved it, and they wanted more. So they said, they wanted me, can you teach a class on synthetic biology? And it's like, okay, I know just enough to be dangerous. I don't know if I could really do this. But again, through ASM has their monthly magazine. At the time, it was called ASM News. And there was this cool article about this woman at MIT, Natalie Kuldell, that was teaching synthetic biology at MIT. And I just, again, my ignorance of, I just shot her an email, said, I'm going to teach a class on synthetic biology. Can you help? And boom, she sent me her course material. She sent me her strains. She just said, here's everything I have. Have fun with it. I was like, what a cool world. What an inspirational person to drive this process. Was fantastic.
So the idea of synthetic biology for newcomers to the program is the idea that you can kind of take things involved with gene expression and make them into modules that you can combine in different ways, usually for a specific goal. For example, I know that some of the iGEM people have put together modules for creating chemicals that have particular aromas.
Mhm.
So you can make a bacterium that smells like a banana.
Mhm. Yeah,
Right? So this
exactly.
idea is a wonderful one. And again, this is why it's called synthetic biology, not that you're making things de novo, but you're using modules. And this is very engineering oriented,
Mhm.
putting modules together to make things happen.
Exactly. And synthetic biology, some say, well, what's the difference between synthetic biology and genetic engineering? And I just kind of synthetic biology is genetic engineering on steroids. You
Yeah.
know, a lot of the early synthetic biologists were electrical engineers. Yeah, this idea of genetic circuits, and they want to know,
Yes.
can we control genetic circuits that way? Absolutely. And so a lot of the stuff. So what Natalie had done is take this then to the K-12 world with her program called BioBuilder, teaching K-12 educators how to introduce synthetic biology in their classroom. It mimics a lot of what we see with, if you're familiar with a program called Project Lead the Way, which is a broad national engineering program to bring engineering into the K-12 education realm. And so Natalie has built this bioengineering, biobuilder program to bring synthetic biology, again, to get more, we have got enough robots. We need more bio stuff. And there's a high school teacher that I first met from Taiwan. He's now in Pennsylvania, I think. That really, how do we do more of this stuff? How do we bring that idea of robotics and stuff like that into the bio world? And so Natalie's taken some great things, taking some of these synthetic biology iGEM projects and putting them in the classroom. It's been fantastic. And so I've been, again, fortunate since Natalie knew me. Some local high school teachers were doing a workshop, so she asked me to help out with them. So I kind of got then involved with helping to spread the word about BioBuilder into the K-12 community. So I've been, again, so fortunate, just building those connections and opening your minds and looking for opportunities have put me in places I couldn't have dreamed of. And so again, very blessed to have those experiences. And so again, working with Natalie and BioBuilder and iGEM, it kind of bring things full circle to where we started with. A big part of iGEM, again, that kind of draws me to it is an important aspect of their competition is what they call human practices. Synthetic biology, should we do synthetic biology, and how do we get it out in the real world to again build dialogue about these things? And so iGEM teams are all about communicating with the general public about synthetic biology and making sure that people understand what we're doing, understand the risks and the safety of the whole process. And so iGEM introduced me to another program that Natalie's been involved with called Building with Biology, which is an outreach program. And so I got our iGEM team together and we got a Building with Biology kit that has all these different activities you can do to introduce synthetic biology. And so then we start going off into the to the schools. I did a great event up at Boys and Girls Club. I bring Girl Scout groups here. I work with a program called Hit the Ground, not Hit the Ground, Expanding Your Horizons for middle school kids. And so I've used this kit in so many different contexts. It's just been phenomenal to introduce them to systemic biology and its potential, but also opening conversations. The conversations I've gotten out of these things. I'll wrap up with one interesting story. When we did one of these events, had a local parent that came to our event and was walking around with her kids and turned to the student and said, "Wait, I thought this was a science event. You're doing genetic engineering. That's not science."
Oh,
And the students were amazing. They sat, they had a dialogue. They must have talked for 45 minutes, going back and forth and just having this great conversation. Maybe not changed the mind, but at least they listened. They heard
yes.
what their concerns were. They explained their side of things, and then there's like, that's the power of outreach. We can't say, oh, you can't do this, you can't do that. We gotta talk to each other. We gotta hear each other. And so that's an event that really has, again, it continues to drive me. Is that it's great to get people excited, it's also great to get that dialogue going back and forth.
And I want to I
cool
want to
stuff.
emphasize this because we really are very concerned with literacy, the be able to read and write, though cursive doesn't seem to exist anymore. We really want to be able to be numerate, the idea of being mathematically literate. I would argue what they really mean is more arithmetic literate, but that's okay.
Yeah.
And then finally, we come to the part where so many times issues revolving or adjacent to science are misunderstood because of different definitions that people have for almost everything. It's not a surprise.
Yeah.
Every language has its own vocabulary. Why wouldn't mathematics have its own vocabulary? Certainly English literature has its own vocabulary, and why not science? So this idea of being able to kind of talk to one another rather than at one another is really critical, and that's the time to do it.
Yeah, that's absolutely right. You know, through building with biology, I've discovered they have these forums you can do. So they've created all the material to talk about genome editing, engineering mosquitoes, and so I've done several events like that. I've done events like that. I've again, my ties in the community. I brought in folks from all over, from the religious community, our international students, getting them all talking together about these topics. And the conversations you get out of this is really, again, so valuable. Again, you hear their perspectives, you understand each other, and they realize that you're not, they understand where you're coming from, that you're not trying to, force these things on people. Just say, we're seeing what's possible and looking for the, just because you can do it doesn't mean it's the right thing to do, but you need to understand what you can do with it to figure it out. So it's been delightful.
Davis, we start to run a little bit short on time. I did want you to talk about something that I think everyone will be interested in hearing more about. Okay. And it revolves around someone who was critical to certainly the development of microbiology, and I think is still very important. Whenever we look at growing bacteria on Petri dishes, we use a thickening agent to solidify the media into its solid form, and we're using a derivative of seaweed called agar. And actually, the person who came up with that idea was...
good old Fanny Hess. Yeah.
And that's right.
Delightful story.
It's a wonderful tale, and I'll put a link in the show notes to it. It's one of the things that I bring up in my microbiology class of kind of names that you ought to know. Yeah. And this is a good one. Because it talks a lot about the development of microbiology. It talks about how the different sexes were thought of, especially, prior to the 21st century, all of that kind of thing. But what's interesting here is you had a really fun way of illustrating Angelina Hessa's portrait. And I thought it would be a good thing to start to close out on when you describe this. You have beautiful photographs, and I think people will be interested to see how they're made.
Absolutely. Yeah. Fanny Hess, again, from my days in Germany, I did my postdoc in Göttingen, Germany, which is the home of Robert Koch. And so that's kind of a tie here. So Fanny Hess, again, one of those misunderstood stories. There's always a story, she was, the wife of his technician and things like that. But that's the time period. She was probably an integral member of that community, incredibly valuable contributions, not just the wife of a technician in the lab. And so when I introduced the history of microbiology, she's a really important figure, because we got to show that women were involved early on. They just don't get the recognition that they deserve. I talk about her and Beatrix Potter and the local microbiologists we had. So I want to make sure that their voices are out there. So Fanny Hess is a big part of that. And recently, I think next year is going to be her 175th anniversary. And there's some colleagues that are putting together a picture book about that. And so through my connections with Natalie Coldell, again, she's drawn some great things. She introduced me to a group at Rice University that had developed a light-sensitive strain of E. coli. It produces a black pigment, but only in the dark. When you expose it to light, the pigment goes away. And so they developed this way to make what they call a bactograph. And so you basically put the bacteria in the lawn of agar. You create a mask, so I can take a photograph of Fanny Hess or anything. I put it on the petri dish, and then I have a setup where I can shine the light through it, kind of like doing a negative when you're developing film. And so shine that through, and then you can get a photograph. And so as you can see in the image that I shared with you, I took a photograph that was shared with me of Fanny Hess, put it on this petri dish, and created her image. Again, a beautiful illustration, very meta, right? Is that the right term? Discover of agar in agar. And again, I have to credit a high school student for getting me to do this. Again, I had a high school student working in my lab, helping me to refine some of these techniques. And we were doing line drawings and stuff like that. He took a picture of Shrek and said, "Can I do this with a picture of Shrek?" And I think there's no way, because it's not, you got to have really fine lines. And it was a photograph. And sure enough, it turned out. It's like, you can make a photograph with this stuff. You get that kind of resolution. And so that, again, moved me along. So it's that student mindset of not, we limit ourselves. Students are unlimited. And we got to, if I end on one thing is my goal is to keep those limits off our students, to let them think wherever they want to go with it. I don't want to be the one to say, no, you can't do that.
Do you know the Mark Twain, Samuel Clemens, the Mark Twain quote, "It's not what you know that gets you in trouble, it's what you know that ain't true."
Absolutely.
And it's and I would add to it something that I talk about first class I teach on anything is so many times students are shut down because they think something is, and I hate this, a stupid question, or an uninformed question, or a fill in the blank, but you want people to be able to question, thoughtfully. And there's an idea from my wasted youth when I was interested in Zen Buddhism called the beginner's mind. And, the mind of an expert is limited, but a beginner's mind isn't. And this is true over and over again. And you just gave a wonderful illustration of exactly that.
Yeah. Just, okay, let's try it.
Yeah. Yep. And that even happened to me in graduate school, where I brought up something to my graduate advisor, would such and such work? And she said, "Oh, not at all. That's really not a great idea." So I said, "Oh, too bad." But the fact is that was correct. We were all wrong in thinking it wouldn't work. So if it's possible to try something, you should. Now, if it costs a lot of money, you have to take that into account. But if it doesn't cost you anything but time, why not?
Yep.
If you think it's worthwhile trying.
Yep. And that's why it's so great to work with students.
Yes.
Because they still have that beginner's mind.
Yeah. So I just wanted to say, as we close out, it's been a real pleasure chatting with you. And I see you every year at ASM, and you've always been such a positive influence to so many people, including me. And I hope that you're kind of the way you are has come across to the listeners and viewers as you being a force for positivity and a force of change. And I want to thank you for being on the program. I wish you and your family the very best.
My pleasure. It's been a delight.
Thanks so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with tasty links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. David Westenberg is part of the Department of Biological Sciences at the Missouri University of Science and Technology in Rolla, Missouri. Many, many thanks to David Renata for superb editing, and Reber Clark for the wonderfully quirky music that brings a smile to my face each week. I hope you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.